Hello everyone, I'm honored to share with you my research for my PhD. My name is Teresa Amponsa and I'm working on a screen of genistein-metabolizing bacteria from pig microbiome. To begin with, my lab is focused on the gut microbiome, be it humans or animals. Now I'm currently working on the pig microbiome. When we talk of gut microbiome, it's basically microorganisms that reside within the gastrointestinal tract. So I'm looking at microorganisms residing in pigs' gastrointestinal tract. Now I'm screening for genistein-metabolizing bacteria. Genistein is actually a phytoestrogen which is structurally similar to estrogen and is being found to be present in soybean. Pigs actually have soybean as part of their feed composition. That means genistein is present in soybean, and there could be some bacteria present in pigs that are able to utilize this genistein. Now, a brief background on what this genistein is. When this compound known as genistein is attached to a sugar molecule, we call it genistein, and once we ingest this genistein, it's being degraded by beta glucosidases, and then the sugar molecule is cleaved off. So we have genistein. So take note of the differences between the spellings. Now this genistein is being metabolized by intestinal bacteria present in the gut microbiome into dihydrogenistein, and finally metabolites are produced. Genistein has 5-hydroxy-ECO as one of its metabolites. So the end product of genistein breakdown is its metabolite known as 5-hydroxy-ECO. Genistein and its metabolite, that is the 5-hydroxy-ECO, both have been shown to have some beneficial effects on the host. So they could be used as antibacterial, anti-proliferative, anti-inflammatory, and even anti-cancerous alternatives. Depending on the geographical location, genistein being present in soybean, so if you belong to a population that takes in a lot of soybean, then you would have a higher amount of genistein present in your system. So most Asian countries ingest soybean. So soybean is actually found in high abundance in Asians. My objectives are first to check how the pig microbiome would change if I introduce genistein, and then once I do this, I would I'm trying to understand how the gut microbiota contributes in generating flavonoid, in my case genistein metabolites, to enhance gut health. And then I would like to test for the antibacterial immune-stimulatory properties of genistein in 2 models. First, I'm going to begin with a mammalian cell culture model, and then I'll follow up with an in vivo germ-free pig model. Now, I forgot to mention that genistein is an isoflavone, and isoflavone forms part of a general group called flavonoids, and these flavonoids are actually abundant in fruits and vegetables, but then isoflavone, being a subclass of the flavonoids, are abundant in soybean. This is my experimental workflow. So I began by collecting fecal samples from the pig farm here in SDSU. I'm a student of South Dakota State University, so I got the fecal samples from our pig farm here. We made sure the pigs were not on any antibiotic use, so they were devoid of antibiotic use. Once I got the fecal samples, I pulled them together and then I ran a bioreactor. Now, the bioreactor is a system where we use in a lab where it's a continuous cell culture model which mimics the hindgut. So we have the media being modified brain heart infusion, and then we supplemented the media with genistein, and the fecal sample of the pigs were treated with the genistein. So basically, we enriched the fecal samples with genistein, and we did this for a period of 21 days in 6 replicates. At the end of the run, I pulled the samples together, that is the genistein treated groups together, and then I sent my samples out to Gout. Now, Gout is actually a micro-cultivation array company that isolates pure cultures from mixed cultures in 96 raw plates. And then once we got the samples back from Gout, I continued with the identification using Malditov mass spectrometry. Now, I also performed anaerobic routine culture methods manually in the lab to try with the identification of bacteria from the pig samples that had been enriched with genistein. Once I got the species, I performed the DPH assay. Now, the DPH assay is actually a fluorescence assay where DPH, which is 1,6-diphenyl 1,3,5-hexatriene, is a fluorescent dye, and when it's bound to genistein would quench its ability to fluoresce. But then when I treat this compound genistein and DPH with my bacteria of interest, if my bacteria is able to break down my genistein or metabolize genistein, then genistein wouldn't quench DPH's fluorescence ability, and then there will be fluorescence. So this assay is basically to test for bacteria that are able to degrade or metabolize genistein. Once I did that, I continued searching for the main purpose is to test for the antibacterial properties of these bacteria that I'm going to that I've isolated from my enrichment. Now, since I want to test for its antibacterial properties, we thought it wise to first be sure that these bacteria are non-pathogenic. So to test for their non-pathogenicity, we performed 2 main screens, one being the hemolysis screening, where we are looking at the ability of my species to break down red blood cells on a sheep blood agar. So if they're able to break down the red blood cells, the sheep blood agar will determine if it's completely hemolytic, partially hemolytic, or non-hemolytic at all. And then we also tested for the ability to invade mammalian cell line, and currently we are still testing for their invasiveness. So these 2 hemolysis screening and invasion assay are just to test for their pathogenicity. And then finally, when we are convinced that the species isolated are non-pathogenic, then we are going to test for their antibacterial properties in vitro cell culture model and in vivo germ-free pig model. Now, this is the outline for the bioreactor that I talked about. So basically, we had 2 groups. The first group is the control, where the fecal samples from the pigs were just inoculated in brain heart infusion media. The treatment group had genistein supplementation of 500 milligrams per liter, and we ran it for a total of 21 days in 6 replicates for both groups. After the samples from gout were received and analyzed for their identification, I saw that most of the species belong to the Streptococcus genera. So you realize that there are 4 species belonging to Streptococcus, that is Streptococcus lutetiensis, Streptococcus equinus, Streptococcus alactolyticus, and Streptococcus galolyticus. That means there was less diversity from the samples received for gout. And then also, we only got just a few species belonging to Acidomycoccus fermentans, Lactobacillus salivarius, Phaecococcus pleomorphos, Peptostreptococcus raceyi, and Mystiocella geloludini. So in total, I got 10 species from gout with less diversity. From my routine isolation, I supplemented my media with 1 milligram per liter of a combination of clindamycin and ampicillin to inhibit the growth of Streptococcus genera, because I did realize that I performed an antibiotic susceptibility screening and I saw that the Streptococcus genera were susceptible to this concentration of ampicillin and clindamycin. So upon inhibition of these species, I was able to isolate other species, that is Enterococcus faecalis, Bacteroides vulgatus, Lactobacillus salivarius, Bacteroides uniformis, Bacteroides flaccus, Lactobacillus agilis, Enterococcus avium. So you could see that there are some there's a relatively high diversity in this routine culture method. This is just after one run of isolation with a gout sample or with the automated system. Out of over 500 samples, I could get only 10 species, but with this manual isolation, out of just one run, I was able to get 6 species. So I was able to get 6 species. So in total, I was able to isolate 15 species. And then upon performing my hemolysis screening, I did realize that 5 of my species were hemolytic. So hemolysis is shown by the clearing of the media. So you see here, Lactobacillus salivarius is able to clear the sheep blood agar. So there's a clear zone on the media. We also saw clearing of Streptococcus alactolyticus, and then Streptococcus equinus and Enterococcus avium as well. Initially, I tested using Bacillus subtilis as a positive control and DH5 alpha as a negative control, but I realized that they were not working as they were not performing their intended purposes. So for my subsequent runs, I stuck to one of my strains, which is Lactobacillus salivarius, which was clearly completely hemolytic, as my positive control. So from this run, I realized that 5 of my species, namely Streptococcus alactolyticus, Streptococcus equinus, Streptococcus Lactobacillus salivarius, Enterococcus avium, and Mystiocella geloludini, were actually hemolytic. So you could see M. geloludini here is also showing some clearance here. Now, please take note that of the 5 species that were hemolytic, only Lactobacillus salivarius was completely hemolytic. That means it was beta hemolytic and it completely cleared the area. The remaining 4 were just partially hemolytic, so they were alpha hemolytic. I tested for the ability to degrade genistein using the DPH assay, and then I saw that of my 10 species, only 2 of them, that is E. coli, E. coli was my negative control, and then Bacillus glycinic fermentans was my positive control. So if it's able to degrade genistein, there's going to be fluorescence and will be visualized as a white patch. So all the white patches you see are showing that they are degrading genistein, and then the brown ones are negative control. That means they are unable to degrade genistein. So the E. coli is a negative control, and you could see that 2 species, that is Peptostreptococcus raceyi and Mystiocella geloludini, are unable to degrade genistein. So in conclusion, I was able to isolate 15 species. Out of these 15 species, 5 were hemolytic, 10 were non-hemolytic, two were unable to degrade genistein, but then I'm going to confirm this with an HPLC analysis to be very sure that this is actually not degrading genistein. And then with my 10 species that were non-hemolytic, I am currently working on the invasion assay. I did the first run, and then I am actually doing a confirmatory run before I'll be convinced of my results. So basically that's what I have for now, and then thank you so much. I'd like to thank Dr. Joyce Carrier and Dr. Sushin Wang, my advisors, as well as Scaria lab members for their immense support throughout this project. Thank you so much.